Okay, so thanks so much for coming today. Good morning. I wanted to introduce myself. I'm Lynn Raymond, professor in psychiatry and medicine, division of neurology, and the director of the Djavad Mowafaghian Centre for Brain Health, your host today. And I have with me Shernaz Bamji, who's our associate director and professor in cellular and physiological sciences. So we'd really like to welcome you warmly to today's event, where we hope to convey our excitement about the future of brain research with presentations from 6 of our top talented scientist leaders who will tell you about some of the highlights in their field. Afterwards, we'll have a more wide-ranging discussion, and we hope to answer your questions during that time. So the Djavad Mowafaghian Centre for Brain Health is dedicated to the principles of truth and reconciliation. We are privileged to work and play on the unceded ancestral lands of the Coast Salish peoples, including territories of the Musqueam, Squamish, and Tsleil-Waututh nations. Sorry about that. What happened? Yep. Yeah, who are we? Skipped a couple slides there. So we are the heart of neuroscience community in British Columbia, not to exclude other universities, but we do have 127 full members who are distributed across all the UBC campuses and represent foundational researchers as well as clinicians. We also host 6 Vancouver Coastal Health clinics in this building, and in 2021 alone, we saw more than 23,000 visits from patients and families. So COVID really illustrated the power of science for us. About 2 months after identifying the first cases, the virus was identified, its DNA was sequenced, and within 10 months, researchers partnered with pharmaceutical industry, offered effective vaccines. But I don't want you to think that all of that progress was really made in only a few months. It was based on foundational research that took place over decades, including from our own, UBC's own, Peter Cullis, who worked on lipid nanoparticles, as well as advances in DNA sequencing and mRNA technologies. So researchers here at DMCBH continue to uncover new understanding of the brain and then work with clinicians to translate those discoveries to better healthcare for our patients. Ways to do this because of the co-location of both foundational researchers and clinicians in the same building, this one that you're in here, the DMCBH. And one reason we're particularly excited about the future of brain research is because of new technologies that are propelling research today, which Shernaz Bamji will tell you about some of them.
Not all of them. So arguably one of the biggest advances in science in the past 20 years is our ability to peer into our genome and to understand our genetic predispositions. So as most of you know, change the slide. As most of you know, the genome is basically a DNA manual for who we are, an organism. Whether that organism is the COVID virus or a fly or a worm or you. It's basically a set of instructions to say to how to create you, your height, your eye color, and so on. And also a set of instructions for how you will function. So do you have a genetic predisposition for heart disease or Alzheimer's disease? Are you genetically predisposed to be able to run really fast or be able to do well in high altitudes and so on? The genome, really, we have a lot of information in our genetic code. So just to kind of give you an example of the advances that we've been able to make, the first human genome was sequenced in 2003. It took 13 years to complete, and it cost $2.7 billion. Just 5 years later, thanks to advances in technology and thanks to foundational science, and you're going to be hearing that word over and over again this morning, thanks to foundational science, we were able to, now it just takes 2 months to sequence the human genome and costs only $1 million. Fast forward to today, it only takes anywhere between 24 hours and a few days to sequence an entire human genome and costs about $600 US. But now comes the fun part or the scary part, which is to kind of mine through this enormous amount of data and to try and make sense of it. So the DMCBH would love to, with some support, hire somebody to be able to do exactly that, work together with artificial intelligence to mine through the genetic data that's already out there in the world, collect more genetic data and mine that from our patients with their, permission, of course, and then to try to make sense of it. So why are we interested in this? 2 words: personalized medicine. So right now, clinical trials are done on large cohorts of patients, and typically patients will fall into 3 different categories. Those that do really well with a given therapy, those that are not affected by the therapy, and those that actually do poorly with a given therapy. Now, unfortunately, even if a given therapy works really well on a subset of patients, it might never see the market, because we don't know right now which patients it'll be good for and which patients it might actually hurt. So it never sees the market. But you can imagine that if we got a lot of information from these patients in the clinical trials, including their genome, their genetic information, then maybe we can find some genetic commonality. So what is in common between the people who do really well with a given therapy? And then identify some sort of genetic signature. So all of these people had this particular thing in common. And so now person X might also do well with that given therapy because they also have that genetic signature. And we're going to be hearing a little bit more about personalized medicine from Dr. Kurt Haas, who's going to be talking about his group's work on autism spectrum disorders and personalized medicine. So now to switch to something a little bit more brain specific. One of the biggest questions in neuroscience, bar none, is what is the function of given regions of the brain? So what are the different areas of the brain and different circuits in the brain are responsible for different human experiences like fear, arousal, anxiety, stress, etc.? You name any given experience, and there is a brain region and a brain circuit that's involved in it. So why is this important to know? Well, if we know what circuits and regions of the brain are involved in these different processes, then we might be able to determine what creates behaviors that are maladaptive. So what parts of the brain are vulnerable to mental health disorders like depression or anxiety or sleep disorders? Now, of course, the goal is not only to identify these regions, but then to eventually try to fix these regions as well, to modulate them, to restore them. And Dr. Fidel Vila-Rodriguez is going to be talking about his work on how to modulate the brain to alleviate depression. But back to, how do we identify the function of different brain regions? Now imagine you're an alien and you see these 3 different machines, these 3 different objects, and you're asked, what is the function of them? Well, just by looking at them, you might surmise some of the function. The object in the middle has got wheels, so it's probably involved in moving. But if you were able to turn on these machines and actually see them in action, it'd be obviously a lot easier to understand the function of these machines. So in the exact same way, researchers have devised a way to selectively turn on certain brain areas, certain brain circuits. So first, we have to genetically modify the neurons that to express a certain chemical that gets activated in response to light. And then all we have to do is shine a light on a particular brain region or a particular brain circuit, and then you can just turn it on and see what it does. So this technology, which is called optogenetics, has given us amazing insight into brain regions, brain circuits that are specialized for a particular function or the other. And so today, you're going to be seeing and hearing a lot more of this kind of cutting-edge technology that UBC researchers are using to really move the needle on some of the largest questions in neuroscience today.
So we're going to hear from our integrated research programs, representatives from each of them. We have 5. You can see them on the slide here. I won't go through the names. And the speakers are shown in the photos and up front. And so you're going to hear about some of the goals of those integrated research programs, which include both foundational sciences, studying things on the molecular cellular level, all the way to the clinic and applying to our patients. You'll hear from them briefly, have a couple of minutes for questions after each talk. Then we'll move to the next one. And when we finish, we will have half an hour to have a broader discussion with you. So we also have some invited scientists from those groups scattered through the audience. I think the scientists have the hanging badges. So they'll be at the lunch afterwards, and you can ask them questions as well. There will be a tour for a small group of people. I'm very sorry, it was a popular option and we were not able to accommodate everyone. So those with the colored dots will be able to go with their groups at 12:15 for a brief tour. So with that, we're going to bring our first speaker up, who unfortunately will have to leave right afterwards because she's giving another talk at a large conference in downtown Vancouver today. This is Dr. Teresa Liu-Ambrose, who is the Canada Research Chair in Physical Activity, Mobility, and Cognitive Neuroscience, and Professor in Rehabilitation Medicine. Teresa?
Thanks, Lynn, and welcome everybody and happy Saturday. It's really great to see all of you here today. So what I like to do now is really to share with all of you some of the research we're currently doing here at UBC, as well as kind of around the world, that really enable each of us to shape our cognitive health as we age. And in turn, really adding life to our years by maintaining our functional independence. So through research, we now actually recognize that there's a number of things we can do that's within our power to actually promote brain health throughout the lifespan. So for instance, we now know that it is really important to maintain a healthy body weight and maintain your blood pressure in midlife. But notably, what I want to highlight is that evidence strongly suggests that engaging in regular physical activity and exercise is key in reducing your risk of developing cognitive impairment and dementia in late life. And that really cognitive impairment is not an inevitable consequence of aging. As such, exercise or regular physical activity is widely recommended now globally as an evidence-based strategy to promoting one's brain health. Nevertheless, there's still a lot of research that needs to be done to achieve the overall goal of using exercise as medicine for healthy cognitive aging. And so what do I mean by that? Well, right now, if you do look at any of the recommendations, they're pretty general. In essence, they recommend something along the lines of you should exercise 2 to 3 times per week, and it may take up to 6 months before you reap the benefits. And that's fine. But again, our goal is to utilize exercise so that it maximizes the benefit at the individual level, so that it's aligned to the individual's preferences, as well as to potentially their physical abilities. And to do that, we really need to understand the what, who, how, and when of exercise prescription, meaning we need to better understand the effects of different types of exercise training, who may benefit more or less. There is some evidence to suggest that not everyone doesn't benefit equally, and we need to understand why that is to tailor. We want to understand how exercise brings about its benefits, and then potentially are there critical times within a lifespan that we really need to intervene to really significantly reduce the risk of dementia in late life. And so today what I want to do is just highlight some key research we have done here at the center within these 4 pillars. And I'll start with the what, which really we have focused on understanding the type of exercise training. So if we had this talk 15 years ago, I'd be pretty limited in what I could share with you, in the sense that I can only talk about the potential benefit of aerobic exercise. So that would be jogging, running, swimming, cycling, anything that increases your heart rate and improves your cardiovascular fitness. Because we simply didn't know anything else. There was very little research in looking at other types of exercise, such as strength training, dance, balance training, yoga, etc. But really, when we think about people adopting exercise as part of their lifestyle, for them to really adhere to it for the long term, we do need to consider, what do you prefer? What do you have access to? And potentially what you're physically able to do. So for people who with limited mobility, perhaps aerobic exercise isn't really the strategy for long-term approach. And so within our research, we have really flagged the potential utility of strength training. And so that's really lifting weights on a regular basis to increase your muscle strength. And what we have shown is that this mode of exercise is also highly beneficial for your cognitive health, can significantly impact your brain, and potentially even reduce existing disease within the brain. And then I'm going to move to now the how. And so understanding how exercise brings about its benefits is complicated, because it's almost kind of like a magical pill or bullet. It has multiple ways by which it can benefit your brain. I don't know if it's just to highlight its complexity, it's actually been shown within the last 5 years, even by contracting your muscle releases a number of proteins or messengers that actually communicates with all other organs in your body, including the brain, that induces additional physiological changes. So it's not just about improving blood flow to the brain as to how exercise brings about its benefits. It's actually quite eloquent, complicated, and fascinating all at once. So within our research, we do tend to apply this multi-pronged, multi-lens approach, whereby when we enroll individuals within our exercise trials, we will scan their brains to better understand how their brain structure or function responds to exercise. We will collect blood to understand what are some of the cellular changes that may occur, as well as measuring their behavior, such as sleep, because we now all recognize that good sleep is essential for a good brain health. And so through our research, we have actually shown multiple responses to exercise at different levels. So level 1, I'm going to refer to as more the cellular and biological response. And through the collection of blood samples, we have shown that with exercise, we actually can see increases in what we call neurotrophic factors circulating within a person's body. And these growth factors really promote the development of new neurons or new brain cells, as well as their survival. And through neuroimaging, we've also managed to show that even after 6 months of exercise training, we can actually see increases in brain volume, specifically in an area called the hippocampus, which is really our primary memory center. So what we have actually shown is after 6 months of training, the hippocampus volume could actually increase by 4%. And that may not sound like a heck of a lot, but to put it in context, even just with natural aging, by the age of 50 and onwards, we actually can see upwards of 1% reduction in hippocampal volume. And that's just with the sort of the natural process of aging, aside from any neurodegenerative diseases that may be occurring. We also have shown that through exercise, you could potentially increase the myelination within the brain, as well as how the brain in itself may communicate with each other. At the level 3 level, which is again related more to behavior, we also have shown that through exercise or after exercise, people's sleep quality improves, which is great, right? Because what we do know is that as we age, the quality of our sleep tends to decline as well. We have a harder time falling asleep. We have a harder time sleeping through the night. And what we have shown is that through exercise, one's sleep quality actually can improve. And again, there's a huge body of literature now supporting the notion of sleep and supporting good brain health. And just to end here, I just want to highlight really some of the impact of our research here at the center. So first and foremost, I really do think our research really has impacted at the global level in highlighting the role of other types of exercise. And I think, again, it's critical. I was trained formally as a physical therapist. And so for me, I use exercise as sort of a strategy for almost everything under the sun. But we do need to recognize, again, people have different preferences, capacities, and access. And so by showing the effects of different types of exercises, we can tailor to the individual. And that is really critically important in, again, long-term adoption of a behavior. Our research also has really informed key reports that's been launched even at the level of the World Health Organization regarding the topic of lifestyle factors and contributing to brain health in later life. And then probably the most meaningful to me is that I do believe also our research has really empowered individuals to feel that they do have some control in how they shape their cognitive health as they age. So thank you.
We have time for a couple of questions. If you have a question, if you're near a mic, please press the button so people can hear you. Dr. Liu-Ambrose will have to leave after this, so any questions about exercise and brain health will be best answered now.
Could you please answer the question regarding the role of dance specifically in maintaining cognitive function?
Yeah, no, I'm really glad you asked about that because that's actually the latest in the news probably in this area. Colleagues of mine in the United States actually just did that. They looked at the role of Brazilian dance. I'm not sure what that actually is in terms of the form of dance, but they study specifically Brazilian dance, and they did actually show benefit. One thing about dance that's really intrigued a lot of us as investigators is that in many ways is a mode of exercise where there is a physical challenge, but also a very much a cognitive challenge as well, right? Because when you're dancing, you're learning new moves, you're coordinating, etc. It's kind of like a complicated, cognitively top-down type of exercise.
Go ahead. Can you press your button and turn off?
How much exercise would an elderly person need to do that would help them to sleep better? My mom is 96 and she really has a hard time sleeping. I don't know how much exercise she could do.
Yeah. So unfortunately, I can't really provide you with a specific regarding the dose, but I would have some recommendations as to the timing. So certainly, I would highly encourage somebody like your mother to engage in, let's say, to do a brisk walk in the morning to kind of really benefit from the natural light in the morning. So that kind of also has an effect on your circadian regulation. What happens as we age is part of the reasons why we sleep have more difficulty in sleeping well is that our circadian clock isn't as well regulated. So it does need additional stimuli to regulate that rhythm. And so natural light is a key driver. And so exposure early in the day would be highly beneficial combined with some physical activity.
Okay, thank you so much, Teresa. Hey, if anyone's standing in the back who is not one of our scientists, there are seats closer to the front, so feel free to move to a seat now if you need to. I'd like to introduce Dr. Mark Cembrowski, who's assistant professor in the Department of Cellular and Physiological Sciences, and he has been awarded the distinction of Next Generation Leader at the Allen Institute for Brain Science in Seattle, Washington. So pleased to introduce Mark.
Our memories make us who we are. When we remember and revisit our past, it's these memories that give us a connection to our current sense of self and our place within our family, within our communities. Our memories are responsible for our imagination, our emotion, our future actions, and in many ways, we are our memories. And perhaps this is nowhere more apparent than for individuals that have memory loss, like that occurring in Alzheimer's disease. For these individuals, losing, pardon me. I believe it's on. I can make it a little. Is this any better, Ricky? I wish it's okay. If it's too quiet, please let me know again, and I'll use the microphone that's up here. Thank you. Okay, thanks. Our memories connect us. Good? Okay, perfect. Thank you very much for your patience. They give us this connection to who we are as individuals and where we are within our communities and in our societies. And in memory loss, one loses not just these past memories and who they were, but also loses their present sense of self and one's place with our families and with our loved ones. Now, what's really striking when we think about the neurobiology of memory is that physically we actually don't know what a memory is in the brain. This is one of the core goals of foundational neuroscience and also holds immense promise to understanding and potentially treating a variety of memory-related ailments like those occurring in Alzheimer's. These broadly define our goals within the Learning, Memories, and Dementias Integrated Research Program here at the Djavad Mowafaghian Centre for Brain Health. Namely, to first understand physically what is a memory, and then to use this knowledge to help us diagnose and treat brain diseases such as dementia. Our Learning, Memory, and Dementias Integrated Research Program has almost 24 researchers that span both foundational and clinical neuroscience. Now, I'll use these terms throughout my talk today, and other speakers will use them today, so I'll quickly tell you exactly what this means. Foundational researchers look at the core building blocks of life, such as DNA, cells, and proteins, and contribute new knowledge on the structure and function of the brain in health and disease. The picture that you can have in your head for these individuals are researchers that are at a university, typically at a bench, much like here at UBC. Clinical researchers alternatively study how the brain changes affect human function, health, and disease as well, but also investigate the effectiveness and safety of new treatments and medications through clinical trials. The picture you can have in your head for these individuals are researchers that typically work in a hospital setting, oftentimes with patients. Finally, clinician scientists conduct work in both the lab and the clinic and are a hybrid of foundational and clinical researchers. Now, within our Learning, Memories, and Dementias Integrated Research Program, we span foundational research, clinical research, and also have a variety of clinical scientists. Some of these individuals that are here today are shown on this slide. These include Dr. Thalia Field, who's a clinician scientist, Dr. Jason Snyder, who's a fundamental neuroscientist, Dr. Manu Madhav, who's a fundamental neuroscientist and classically trained as an engineer, and myself, who's also a foundational neuroscientist, but I'm classically trained as a mathematician. Together, we combine all of these different disciplines to go after one core goal together within our program, to understand the neuroscientific basis of learning and memory, and to use this knowledge to prevent and treat dementias. As Dr. Bamji alluded to earlier today, we're really in the golden age of using technology to understand the brain. And with this technology, I think we're exceptionally well poised to address one of the key societal questions in today's age. Our big question is, how can we understand, treat, and potentially prevent Alzheimer's disease? Now, obviously, this is an incredibly complicated question that requires a variety of expertise spanning clinical neuroscience and foundational neuroscience, but it should also involve a lot of other disciplines. These disciplines can include those individuals that develop tools and can apply these next generation tools. What I'll show you over the last few minutes here in my talk today is a collaboration going on here at UBC that spans all of these different aspects. That starts with brain surgeons and neuropathologists. From there moves to foundational neuroscientists, biomedical engineers, and mathematicians. And what's really unique about this research program that I'll show you here today is that it uses living human brain tissue. We're one of the few places in the world that are capable of doing this type of research, and I'm really excited to share this research with you here today. So this research program begins with brain surgeons at Vancouver General Hospital, about 15 minutes down the road here. These brain surgeons are world-renowned and perform surgeries for those that suffer from epilepsy. The idea being that the parts of the brain that are responsible for seizures can be identified and therapeutically removed, and it leads to really wonderful patient outcomes. Now, typically in the context of these removed brain regions, they're given to neuropathologists that are also at Vancouver General Hospital who inspect this tissue, but the tissue is not used further. What we've been really fortunate to do here is to work with these brain surgeons and to work with these neuropathologists to very quickly receive this living human brain tissue. In the span of just a few minutes, it goes from the operating room into one of our beakers that contains artificial cerebral spinal fluid. This is the fluid that would normally mimic what's present in the brain. Remarkably, this keeps the tissue alive. And although, of course, there's nothing like consciousness going on this block of tissue that we receive, there is a precise opportunity to understand the organization and operation of the living human brain. We are immensely grateful to all of our participants that give informed consent to do these types of procedures. Without them, our research program would not go forward. So we're immensely thankful to those individuals. What we've been able to do here at UBC is to receive this tissue. We've been able to figure out tricks to keep it alive for up to months at a time, allowing us to do a variety of different types of fundamental neuroscience experiments, and also use this tissue for driving new insight using biomedical engineering and mathematics. One of the technologies that we're working on and designing and implementing here at UBC is shown here on this slide. What I'm depicting here is a slice of tissue from the brain. When one opens up a textbook and reads about the cells that are present in the brain, there are some cells that are called neurons. There are other non-neuronal cells like astrocytes here. What's really striking is that we probably appreciate the human brain cannot be this simple. There's got to be a lot of molecular and cellular complexity to this tissue. Using this tissue, we're able to identify methods using sequencing that can take this tissue and identify very subtle relationships. What I'm showing you here, individual points are individual cells from this tissue. They're colored according to the cell type that they come from. And when they're close by to each other, this represents groups of cells that have similar properties. We can take these cells and move back and forth, find out where they sit originally in the tissue, and then use this knowledge to appreciate things like just fundamentally what is the organization of the human brain, and what are the cells and what are the molecules that are dysregulated in disorders such as Alzheimer's disease. These types of experiments allow us to quickly pinpoint candidate cells, genes, proteins that we can target for next generation therapeutics. Now, to actually assess drugs that can intervene and potentially have some therapeutic efficacy, we use this living human brain tissue, and we can keep it alive for weeks using something that's called cell culture, shown here. Being able to use this tissue for weeks allows us to use things that are broadly called designer viruses. These viruses, which are made commercially, can be used to label, manipulate specific neurons, and we can directly observe specific molecules and cells in the living human brain. One example of these sorts of experiments is shown here. What I'm showing you is this is a section of living human brain, and we've used these designer viruses to cause neurons to change color in response to drug applications. What this allows us to do is optically, with a microscope, apply drugs and see how activity in the human brain changes. It's with these sorts of next generation technologies, this allows us to have precise monitoring and disruption of pathologies in Alzheimer's disease to identify new targets that we can pursue and then actively and directly pursue them in these experiments here. So to broadly sum up, I'll give you our vision. I began with this question of how do we understand, treat, and potentially prevent Alzheimer's disease. I've explained to you two limbs of some of the research that's being done within our Learning Memories and Dementias research program here, where we're developing and applying technologies to allow us to identify candidate targets that we can pursue therapeutically, and then to use this knowledge and directly test drug interventions, both designing new drugs as well as repurposing existing drugs to help target some of these candidates that we see in our experiments, and hopefully in the long run be able to use these in a clinical setting. Our ultimate goal, of course, is to use these results to identify new approaches for tackling Alzheimer's disease. HAOS, of course, is just one research program of many that we have going on in our Learning, Memory, and Dementias group, And I'm happy to discuss this in particular or any questions that you may have. And thank you for being here this morning.
So maybe one question, and then in the interest of time, we're going to move on. Alzheimer's is a huge topic, I'm sure a lot of interest, and we will have time to have a wider discussion later, but go ahead, you're our question.
You're talking about Alzheimer's. I have Parkinson's, and I have a memory problem immediately. If I don't take my medication when my alarm goes off, immediately it's gone. If I decide I need something out of across the kitchen, by the time I walk over to the cupboard, I've forgotten what it is. But beyond that, everything's perfect. I can remember everything back to my childhood days. And is that separate research, or is that something you're looking at too?
It would complement some of the work that we're doing. Our work that we're analyzing here, the brain regions that we receive are typically the hippocampus, which is involved primarily in learning memory, and as well Alzheimer's disease. Oftentimes Parkinson's is other brain regions, but it may involve interplays with the hippocampus as well. So I think by understanding our work here in the hippocampus, this gives us a very important foundation with which we can interpret hippocampal disorders in other types of settings, like for Parkinson's. Thank you.
Thanks. Maybe turn off the microphone there. That's great. I'm going to introduce our next speaker. This is Dr. Cheryl Wellington, Professor and Vice Chair of Research for the Department of Pathology and Laboratory Medicine. She's also an investigator for ICORD, which is International Collaboration on Repair Discoveries at VGH, or Vancouver General Hospital. I think she'll mention that as well.
Thanks so much, Lynn, and thanks to all of you for spending your Saturday morning here. It is such an honor to be able to share the work that we're doing with all of you. I have the privilege today of representing the Brain Injury and Repair Integrated Research Program. Accidents happen. You can get a concussion, you can have a stroke, you can have a motor vehicle accident, you can fall. Brain injuries can happen to anyone at any time, and they do impact downstream effects. So for example, we know a brain injury can alter your risk for dementia or cognitive decline. So what our group is doing is looking at how neurological injuries, what happens in the acutely injured brain, how can we help repair the acutely injured brain, and how can we optimize the further quality of life for people that do sustain a brain injury or other acute neurological insult. Because we really don't know much about what happens in the injured brain, nor how to treat it very effectively. So our group consists of around 25 members, again, spanning the foundational, clinical, and translational groups that work on basic and foundational and clinical studies on neurotrauma across a wide variety of neurological insults. I'm going to tell you two vignettes that showcase just some of the work that we are doing. The first one investigates the work that we're doing in the critical care setting. And this is with a collaboration by an amazing intensivist by the name of Dr. Mipinder Sekon. He trained in British Columbia and then also in the UK to develop some very sophisticated techniques for monitoring the acutely injured brain. And he was really the one that began the program of using what we call a brain bolt to understand and monitor a patient with a very acute and serious brain injury. One of the major projects that he has is understanding how the brain is impacted after cardiac arrest. So if your heart stops, could be from a heart attack, it could be for younger people, very often these days it's a drug overdose. If the heart is stopping, there's no blood that is pumping through the body and including the brain. And because the brain is the organ that consumes 25% of each heartbeat's worth of blood and oxygen, it is very vulnerable to injury. So even in patients where we can get the heart restarted, It's the neurological recovery that really dictates how well they are going to do. And so we're trying to use very sophisticated methods to understand this. And one of these is this amazing brain bolt technology. So through surgical insertion of this bolt, we can monitor the pressure inside the brain. We can monitor how much oxygen is getting into the brain tissue. And I'm going to show you some really amazing new research in this area, because getting oxygen back into that brain tissue is one of the primary drivers of either a good or a poor outcome. So the new research that I have the pleasure to share with you today is that we can now begin to use a blood test to get the same information as this very invasive, expensive, and resource-limited brain bolt technology. Imagine that. 10 years ago, we would never imagine that we could do a blood test to look at this, but we can now. And the program that we have on developing blood tests spans a lot of conditions in neurology. We support many, many programs. But what we're showing here, so what's very cool about, we can take a blood sample and we can use a machine that is as sensitive as measuring 1 grain of sugar in an Olympic-sized swimming pool. Wow, that and we are the first. DMCBH was the first in Canada to acquire this technology. So by using this technology, we can measure there on the graph on the left-hand side, there's a protein called GFAP, and on the right-hand side called NFL, or neurofilament light. The gray bars are healthy people without cardiac arrest. So we can measure what is the normal levels there. The pink bars are patients that had a cardiac arrest, heart got started, and the brain bolt is telling us that they're getting good oxygenation back into the brain. And we can see their levels are a little bit elevated, but they're not nearly as high as the levels in patients who, even if we're getting the heart started, somehow the oxygen is not crossing into the brain properly. And these are the patients that have a poorer outcome. What this technology is now going to enable us to do is to have far more personalized care, to be able to monitor almost in real time from blood samples that we can take every couple of hours. How is that brain doing? And how can we adjust the perfusion pressure, the amount of oxygen, having a transfusion given to that patient? Can we do better at getting oxygen back into that brain, enabling that person to have the optimal quality of life? So this is one major program that we're doing. The other one that I would really like to highlight is a program in spinal cord injury led by Dr. Brian Kwan, who is an internationally regarded leader and innovator in spinal cord injury, and who runs many, many programs out of the I-Cord Center near VGH. And here with spinal cord injury, we also found that we can use blood tests to more accurately classify the severity of a spinal cord injury. And what is really exciting, I'll explain these, so don't get worried that these are really complicated graphs, but we can use these blood tests to help predict which patients are going to get motor function back 6 months later. So what you're seeing here, the orange and the blue, all you really need to know or see is that the orange and the blue dots are different. The patients who do well, who regain motor recovery, are in the blue. And you can see that their levels of biomarkers, these brain biomarkers, are lower. And they tend to, for the one on the bottom, it goes down over time after the first 4 days after a spinal cord injury. So we can use the blood test to kind of guess or try to predict which patients are going to recover to some extent and which patients are just not. And this is a graph that kind of tells us our scorecard. How often do we get it right? And if you look at the numbers on the very right-hand side, you see numbers like 0.08, 0.9. This is telling us that we are, by using these blood tests, we're about 80% or 90% accurate in getting it right and predicting how these patients are going to do 6 months down the line. So these sorts of technologies, these sorts of research studies are now helping us reshape how we can do clinical trials in acute neurological indications. So we can have better predictive value. We can have a better way to measure whether an intervention is effective for acute brain disorders, and really help a lot of patients who have an accident that is a life-altering event. And we then intersect these programs with the ones that Mark had talked about and Teresa talked about in terms of their long-term risk for cognitive decline and dementia. So really what I'm very proud to say is that the DMCVH has extensive experience and interest and expertise in acute neurological injuries. These span many different types: concussion, traumatic brain injury, the hypoxic ischemic brain injury after cardiac arrest that I spoke about a little bit, spinal cord injury, and many, many, many others. So we are working very collaboratively across this whole team to try to understand how do we make sure that we can restore as much function to that injured brain as possible at any point in the lifespan, from children all the way up to people in their 80s and 90s. So that's what we're trying to do. And the programs that I shared in terms of these blood tests are also being leveraged into all of the other things. We have programs doing blood tests in multiple sclerosis and in dementia, et cetera, et cetera. So we are one of the core platforms that integrates across the different groups that you're hearing about today as well. And I wanted to mention that because I think it highlights the very interdisciplinary and collaborative nature of the whole center. So I'm happy to.
We have time for one question and then more at the end.
Hi. I'm not, oh, it's on.
Yeah.
I'm interested to know if you have thought about, or do you use hyperbaric in the initial phases of an acute injury?
We have not, to my knowledge. I know it's being used a lot in sport-related concussion work. I am not aware of research here that is using that as a strategy. But certainly, one of the challenges, or one of the benefits potentially of using the hyperbaric oxygen, is that there's more oxygen to try to get into the brain. Sometimes, though, even though there might be more oxygen, the problem is getting it into the brain. So that's where the real heavy lifting still needs to occur. But thank you for the question.
Thank you. Our next speaker is Dr. Fidel Vila-Rodriguez, Assistant Professor, Department of Psychiatry, and he's also the director of the, you can tell me, the neurostimulation and imaging.
It'll give me an opportunity to test the sound. The non-invasive neurostimulation therapies lab. Sound okay? Great. Thanks so much, Lynn, and thanks so much also Shernaz for putting together this great event. And it's an honor to share with colleagues and to represent, in my case, the Mental Health and Addictions Integrated Research Program. I'm just the face today, but I'm really showcasing the work of many. And the theme of integrating as well with other colleagues will be pervasive through the talk. Just going to reach for these slides. And I'm a clinician scientist, and Mark earlier provided you a hint of what that means. And the work that I'm going to share with you wants to tackle one of the grand challenges that we face worldwide. Mental illness is the leading cause of disability in the world. We all know someone close to us, or even ourselves at some point in our life, we're going to struggle with a mental illness. It's really, really common. And neuroscience and clinical research, I think we're living in a very exciting time when they're starting to meet, where they're going to start to really provide solutions to this grand challenge. And the integrated research program in mental health and addictions is composed by 13 of those foundational researchers, a basic science, 6 clinical researchers, and 9 clinician scientists. I'm going to tell you about one particular example that tells a story about translation of research into the clinic. But that's just one of an example of many that happens here in this building. And people, and you're going to be hearing now and later. I want to just quickly say hi to Dr. van der Waal there, who is part of the integrated research program, works in youth, and Dr. Christian Schutz, right there, who works in addictions. And so feel free to tap their brains on their areas of research later on. Again, the fundamental aspect that we want and we are accomplishing here in this center is to bring people to work together and to bridge that gap between research, understanding how things work, how the brain works, to translate and to make treatments, interventions that help diagnose, like Dr. Wellington just mentioned, or that allow to treat or prevent illnesses. So we are all focused on this very goal. And in the integrated research program in mental health and addictions, we're focused on this particular niche of neuropsychiatric conditions. The case that I'm going to share with you is about a technology that allows us to treat treatment-resistant depression, which is a very common condition. And the technology is called transcranial magnetic stimulation, or TMS. I'm going to see, there it is. So what you can see on the screen is a real kind of TMS device here. It's a big box and has attached this kind of wand, but it's a coil. And this coil allows us to stimulate a very precise area in the brain. How does that happen? I just brought a mini TMS device here that I'm going to demo quickly because it still fascinates me how leveraging a physics principle like electromagnetic induction can be used to treat depression and other psychiatric conditions. So we're going to dim the lights up just a little bit so that you can see the little device here. And this is the equivalent of the TMS device. It has a source of energy, 3 AA batteries. That's the plug. It has a really powerful computer, this little piece of electronic here, that generates a type of current that goes through this loop of wires, which is the coil that I have in my hand on the picture. There's a little switch button here that if I press, shines a little LED light here. It means that current is flowing. It's hard to see, but current is flowing. And we don't see anything here, but you have to picture a donut, 3-dimensional donut, literally a donut, yummy, here. Now, where is the brain? Thankfully, I always carry with me a second brain. Mine is not enough. So here's a model of a brain, very simplified, is a loop of wires. And if this loop of wires aligns with that donut, when energy is flowing through, it should induce current and the light should go on. Drum roll. All right. One time it didn't work. All right. Ha. This is what we do with TMS. We shine light in dark places in the brain. Play with it, pass it around. It's a lot of fun. There's no risk. It's very tiny little energy, so nothing's going to happen to anyone. It's not going to help for treatment, that one. How we translated this fascinating technology into treatment. What happened here was we were part of a clinical trial that investigated the use of TMS to treat depression. And the trial asked the question as to whether a new form of this treatment that only takes 3 minutes to deliver, given every day, Monday to Friday, for 30 days, could treat treatment-resistant depression. And the answer of that trial was yes, indeed, it does help. We found that between 50 to 55% of people, their symptoms of depression decreased by at least half, and there were 30 to 35% of them who went into remission, which is what we use to exemplify when there's barely no symptoms or no symptoms left. that was all great and good. We did the science, but what we needed to do next was to persuade the authorities to implement this into clinical practice. 9 months after reporting on this trial, the FDA in the US approved this particular protocol called intermittent theta burst stimulation to be implemented in clinical practice. And last January 2022, the government of BC recommended TMS to be implemented in the healthcare system in BC. That is, well, thanks. And I'm not sure if I'm going to ever see this again in my career. It's really exceptional. But this is really what happens, what we are all focused on enabling. And I think it's a very good story in terms of how that science, going from basic understanding of the brain all the way to clinic, works. Again, that's all good and fine. We've got the technology, but what really matters, that what really motivates all of us are the people, are the outcomes, are changing lives for better. So one of the things that I really enjoy about doing clinical research is that we have that immediate reward, that situation where we see people after 6 weeks, gosh, they're getting better. The TMS technicians that deliver the treatment tell me they can notice, oh, this person is doing better. They are talking. They look happier. That is what we are going after. That's the challenge that we face, and that's what we will put all our focus and attention to make it happen. So thanks for joining the Spark.
Okay, fantastic demo. One question, there's somebody right there who really wants to ask one.
I have a question, sorry.
I'm going to turn this off.
Okay. So your TMS technology, fantastic. What I'd like to understand is it often thought of as a replacement or an adjunct to ECT?
Here, do you want to use this one?
That's an excellent question. And whenever I talk about generating an electric field in the brain with TMS, the question, oh, how is this similar to ECT comes up, because it's also a non-invasive neurostimulation technique that treats depression. The physics of those two treatments are very different. With ECT, what we want to do is to trigger a very short and controlled seizure for treatment. With TMS, we do not trigger seizures. The person is fully awake, as you saw in the treatment chair, receives the treatment, and we don't trigger a convulsion, a seizure. So neurostimulation, non-invasive neurostimulation techniques, but different mechanisms by which they work.
All right, thank you so much.
Thank you.
Next, we have Dr. Kurt Haas, who's professor in the Department of Cellular and Physiological Sciences, and also a founding member of the New School for Biomedical Engineering. Kurt.
Thanks, Lynn. Can you hear me? Yes. Thanks. Yeah, well, thank you for coming. It's a really valuable interaction for us to be able to explain what we do and to hear from you about what you're interested in, what your concerns are, what your needs are. So I had this group that it's the Brain Development in Neurodevelopmental Disorders group. And this is really an incredible challenge to understand how the brain develops. We've been hearing about the wonders of the human brain. We have to realize that this is an organ that is comprised of something on the order of 86 billion neurons. These are these weird long neurons that have these long processes of dendrites and axons that interconnect with each other. So they form on the order of trillions of synapses. So it's one of the most complex elements or objects in the universe. And by itself, it's amazing. But when we think about how it forms, it's rather incredible. How do you develop such a thing? And we think that while most of us are reasonably functional, errors during development, during this very complex process, is what gives rise to neurodevelopmental disorders. These can be quite common. We're very interested in autism spectrum disorders, which is increasing in its prevalence, up to about 1 in every 60 kids today, as well as things like schizophrenia and epilepsy. Unfortunately, there are very few treatments for any of these disorders. There are no effective treatments currently for autism. And we think that a large reason for that is what Shernaz Bamji alluded to in the beginning, is that people, all of us are different. Autism particularly is called autism spectrum disorder because people vary so much in their presentation, their cognitive abilities, their ability to communicate. And we think that the underlying cause is variable as well, and that it requires a personalized medicine approach. And that we've heard that if you don't take a personalized medicine approach and you combine all of your people together when you're doing a drug trial, you won't fix all problems, and you throw those drugs away. So our group is, which is made up of 19 foundational research scientists, partnering with 3 clinical geneticists, along with international partners. Our goals are really to understand this personalized medicine problem. And we take a genetic viewpoint of it. There are other reasons why you might have a personalized need for medicine or outcomes. And Cheryl mentioned some of these. You might have a biomarker for something, but a major driver, we think, is your genetics. So we're very focused on understanding the genetics of brain development and the genetics of neurodevelopmental disorders, leading, we think, ultimately to a personalized medicine approach. But this is it's a long-term problem. So Dr. Bamji, in her intro, raised this point, that one of the most amazing achievements by humans was the sequencing of our genome. And this was done back in 2003. It was a monumental effort. I see it as important as putting a man on the moon. And its impact on science and medicine has that huge impact, because we can finally link genes, our genetic code, to disease states. And this is really amazing. It costs a lot. And as Dr. Bamji mentioned, that investment not just gave us that code, but it also fomented the race to increase the pace of sequencing and decreasing the costs. And that's resulted in projects like the 100,000 Genome Project. So where the large numbers of people are now sequenced, something on the order of hundreds of thousands. And you might say, why bother? We've already sequenced the human genome. We know the code. We know what it takes to build a human. And their answer is, when we started sequencing people, we saw something that was quite remarkable as well. And that is that all of our genomes are different. We all vary. If you look to your left and your right, you vary from your neighbor in your genome, your code, in 1 out of every 1,000 bases. So, and the question is, what does that mean? And is that the basis for our individuality, including the individuality for our disease susceptibility, our response to treatments? And that's what we're here to investigate. So where do these variations in our code come from? Why does somebody have, in this case, an A versus a G in their nucleotide base point in that part of your code? And the answer is from replication, or when the DNA is copied. Every time a cell divides, you have to copy your DNA for the daughter cells. When you make eggs or sperm for procreation, you have to copy your DNA. And even though the biology underlying this replication is fantastic, the fidelity is amazing, errors creep in. And there's something on the order of about 200 mistakes per the entire genome, which is 3.2 billion nucleotides. So it's not very much, but that means in our offspring, there are these slight differences, and over generations, those variants accumulate, creating all the variation within us. So why does that matter? Why do gene variations matter? It's because genes in our DNA variants turn into protein variants. The way that it works is that your DNA is transcribed into RNA, which is then translated into proteins that are the building blocks that make up our entire bodies. Proteins are really just chains of amino acids, and the code from your DNA turns into codes of amino acids. So if you have a change in your DNA code, you have a change in your amino acid code. Proteins typically have hundreds, maybe thousands of amino acid blocks building them up. So you might say, why does it matter that you change one amino acid from one to another? Does it really matter? We're beginning to learn, this is a sort of forefront of the science, that these little changes can have profound impacts on our disease susceptibility and our response to treatments. So this is what our need is. This is what our group does. We try to look at individuals, both the normal population and disease-associated peoples, and to sequence them, find their genetic variants, see how that impacts their proteins, and with doing so, build up this foundation for personalized medicine. So this is the workflow that we do, and I'll sort of step through this. This is what we do for a large group of both foundational and clinical researchers. We start with the patients. So if you think about autism spectrum disorder, again, these individuals have a wide range of impacts on their functionality. So that has to be characterized. So our clinical researchers assess these individuals, they describe their phenotypes, and then they sequence them. These are medical geneticists. They sequence both the patients and ideally the parents as well to find these slight variants in their DNA. And then they hand off those big lists of variants and they give them to our foundational scientists where they make physical DNA libraries. They'll take the same gene that encodes a protein and they'll make hundreds of different variants of it with single patient-derived variants of those DNAs. And that's a laborious, time-producing, costly thing to do, but it's very important. It creates this physical library. And then we take those DNA libraries, we put them into cells where they're made into proteins. And then we can test those proteins to say, is there an impact of this one amino acid change on protein function? And this is really the hard work. We have to develop assays. We have to do, it's costly and time consuming to taste one protein at a time, We do this for hundreds of cases. What I'm showing here in the bottom is the rotating structure is actually a protein called P10, which is one of the leading candidates for autism. There are many genes associated. P10 is a major one. And we've recently completed a study where we tested 127 variants of P10 in 18 assays in 5 model systems. It's what we call deep phenotypic profiling. And we do that because it's very hard to really test all the functions of protein. They're very complex. So we need a lot of assays to do it. And the color bits on this rotating structure are individual amino acids that we have changed, and we've highlighted them with different colors to which ones we think have different properties. And we think that they cause different dysfunctions to this one protein that will likely have to be treated with different types of therapeutic approaches. So in this case, in many cases, we think that a disease is not just caused by, it's caused by a gene, but it's caused by different mutations on those genes, leading to different disease outcome, presentation, different disease presentations, and different response to treatments. So once we get through this stage, which is laborious, we hand off these genes as well to our researchers that study what happens to neural development. So a lot of this work is done in cell culture, some in animals. What you're looking at are actually beautiful pictures of neurons. These are brain neurons and these are experiments we can look at both the growth of their long processes as well as their formation of these synaptic connections between cells, which are labeled here in the purple and the blue in the middle pictures. But through very careful studies, we can see how the same variant or the protein with different variants impacts neuron growth and connectivity, eventually building up our model for how this impacts an organism in the human brain. So where do we go from here? Well, our group has what we call functionalized or tested on the order of 500 gene variants of more than 10 autism-associated genes. And a big part of this project has been developing the platforms to study this. Things move faster and faster. These numbers increase every week. But we're at the beginning. Autism particularly is not a disease caused by one gene. There are more than 100 genes that are tightly linked to autism, and probably something on the order of 600 more that are loosely associated with it. Each one of those genes has tens to hundreds of variants found in individuals with autism that we have to test. So we have a lot of work to go to test this. And then what's the next stage? The most important thing is to develop this into therapeutics. So now that we've developed our platforms, we can turn them on their heads and not just say, use those platforms to find protein dysfunction, but we can use them for drug discovery to see how can we fix those dysfunctions and find novel therapeutics specific to the dysfunction that's there. This is, again, personalized medicine. So our outcomes are this. By finding the genetic cause of a disorder, provides information to families and their patients. This is very important for moving forward for families if they want to do family planning. They want to know if they have risk for future problems. And it's very important for grouping individuals with autism into different groups that might benefit from different treatment options. This empowers our patients. It importantly helps us to develop new diagnostic tools for faster diagnosis for finding problems, but critically, this is the foundation for developing new drug therapeutics for personalized medicine. So with that, I'll take a question.
Okay, we have time for one quick question, and we're almost done, so there'll be plenty of time to discuss. Go ahead.
I'm just wondering on your research if it includes SHANK3.
Yes, SHANK3 is a very important autism-associated protein that we're definitely looking at. Yes. Thank you.
Maybe Kurt can tell you more about that in our discussion. You can bring it up again. Thanks so much. Our last speaker is Dr. Silke Cresswell, and she is Professor, Department of Medicine, Division of Neurology, and the Director of the Pacific Parkinson's Research Group, or Center, rather.
Thank you. Good morning. It's so lovely to see so many of you sharing this passion for the brain. And I'm going to introduce you to the sensory motor and movement disorder group. It's a large group, and it looks at how the brain determines pretty much any sensory input, looks at motor output, how do we experience the world, and also how do we actually move in this world. We want to understand both the cellular and as well as the brain circuitry underlying those sensory and motor disorders that can occur when things go wrong. We want to, and we do, conduct clinical trials and test novel therapies. And we want to improve the diagnosis and the treatment, and very importantly, the quality of life of people living with those disorders. And these disorders are Huntington's disease, so this is something that Dr. Lynn Raymond is working on. It is amyotrophic lateral sclerosis, Lou Gehrig's disease. It is multiple sclerosis. It is Parkinson's disease and other disorders as well. Our investigators, we have, as I said, a large group. We have 16 foundational researchers. We've got 4 clinical researchers and 12 clinician scientists. And if you want to ask a few people of this in this group about their work, we have several of them here. We've got Dr. Jacqueline Quant. Where is she now? Oh, there she's in the back. She is one of the super experts in the immunology of multiple sclerosis. Then we've got Dr. Doug Altschuler. Where is he? So I don't see him right now, but if you always wanted to know why birds can fly, he's the person to speak to. Dr. Lynn Raymond, you already know, for the Huntington's disease. And then Dr. Martin McCune at the back, Parkinson's expert, also an engineer neurologist, and expert for also non-invasive neuromodulation. I'm going to take you to the example of Parkinson's disease, because that's where I work as well. And when you look at this map, there is a very suspicious, very dark red area on there, and it is Canada. And this is a map of the prevalence, so the frequency of Parkinson's disease in the world. And Canada is unfortunately the darkest red on this whole map. So according to the study, which was published in one of the premier journals in neurology, we have the biggest problem in our country. Now, this might be because this is, of course, several studies that are represented there. This might be because we're particularly good at picking it up. But it might also hint to an actual problem, and we don't quite know yet why Canada would feature so heavily there. Now, one of the things that Parkinson's goes with is aging. As we all get older and also live longer, we have more disorders that are associated with aging, including Parkinson's disease. But there's also a correlation with industrialization. And what I'll talk to you in a little bit is to look at what are the changes in our environment that might come with industrialization and our change in the way we live. I also want to take you outside the brain. We've been talking about the brain a lot, but the brain, of course, doesn't live in a vacuum. It is supported by the whole organism, and it's influenced by the environment. And it turns out that Parkinson's probably is quite closely related to the gut. And if you look at the clinical history, and I'm a clinician as well, and you ask people what happened first, and you look at big cohort studies, it turns out that for at least a good portion of people, and that speaks to the personalization of treatments again, the first problem wasn't something that was obviously related to the brain. The first problem was constipation, maybe even 20 years before Parkinson's symptoms ever occurred. So constipation can be a very early symptom. And with that information and a few other pieces of information that I'll skip right now, just because I know we're running late and there's lunch, of course, the microbes that live in the gut microbiome, seem to have a role playing in Parkinson's disease. And to make this very long story short, we have a large study here. We've got over 300 people whom we've been studying for 5 years now, who've given us samples of their stool and blood samples, and who filled in very generously a whole lot of questionnaires. So putting that whole story together, it turns out that what happens is, and you see the there we go. Let's see whether the pointer works. There we go. I talked about the constipation that seems to be a very early feature of Parkinson's. And with that, there's prolonged transit time. Everything moves through the gut much more slowly. And you can also get irritable bowel symptoms with that. That then in itself can lead to changes in the gut microbiome. You get bacteria that are less favorable, bacteria that are producing more toxic metabolites, bacteria that are producing less short-chain fatty acids, and short-chain fatty acids are really valuable both at the level of the gut, where they reduce inflammation and they help us have a good, healthy gut lining. And then we get increased local inflammation at the level of the gut, which is present in Parkinson's. And then we get damage to the lining of the gut, we get leaky gut. Now, all of that then leads to more misfunction of the gut, which leads to more constipation, more irritability, and more prolonged transit time. And then we have a perfect vicious circle going. And that then affects the brain as well, because you get systemic inflammation, low-grade systemic inflammation. You might have heard of inflammaging. And that then has an effect, in all likelihood, on the brain as well, with also inflammatory changes. But we've also, for example, shown that toxins that are produced by the gut bacteria can be shown at higher levels in the blood of people with Parkinson's, and that same toxin has been found in the cerebrospinal fluid. So coming back to what influences our gut microbiome, and it's lunchtime, and I have a feeling this was on purpose to place this talk now. when we look at industrialization, we often move towards a more westernized diet. And this picture, I took this in the checkout line at a big store that is probably well known to you and me. I had just bought blueberries and nuts, may I just add this. But so there we're standing and I thought, wow, this is the epitome of the worst of the Western diet here. Not even sure it qualifies as food, but there we go. So compare that to this. And that's a hard one to show at lunchtime because, that is pretty appetizing. It turns out that a healthy diet, and I'm putting the example of the Mind diet up here, which is one of the Mediterranean diets. So based on lots of fresh fruit and vegetables, olive oil, nuts, legumes, healthy whole grains, etc., and cut out, at least mostly, red meat and fried food and processed food and lots of sugary drinks and pastries en masse. So a healthy diet actually is associated, and we found this here in the same cohort that we studied the microbiome in the gut in, because diet is so important for the microbiome. These participants also very generously filled in very detailed diet questionnaires. And it turns out that if you eat a healthy diet that correlates with the age of onset of Parkinson's, this is up to 8 to 10 years for men, that the age of onset is higher, and up to 17 years for women. That is rather remarkable if you think about the fact that we don't have any treatments, no medications that would cure the disease, reverse the disease, delay the disease, etc. So diet really seems to play a big role. And in the meantime, then after we published that, there was a big study with over 120,000 people that were followed in the US for over 30 years, looking at many disorders, but including Parkinson's. And I won't go into the details. The takeaway point is that the people who were eating a healthy diet, top quartile, exercising, and also top quartile of exercise participation, and then they looked at the people who developed Parkinson's. Those who are leading a healthy lifestyle were 65%, so essentially 2/3 less likely to die in the follow-up period of the study. Now, that is amazing to me, and obviously you have positive impact on a whole range of other conditions, diabetes, cardiovascular health, obesity, cancers, etc. So what is the future putting this together, the gut microbiome as well as the diet? Well, a lot, I would say. We are already working on probiotics, so specific bacteria that we can give as a little capsule to influence the function of not just the gut, but also of the brain. We've got 2 studies going on right now. One is looking at probiotics in the treatment of anxiety in Parkinson's. Another one that is about to start on depression in Parkinson's disease. We can look at things like diet to influence gut health, and we're looking at ketogenic and Mediterranean diets. We can do a combined intervention, and this is something we're also about to start in Healthy Ages. It's called Impact 360 for Healthy Ages. So if anybody's interested, let me know, where we're going to test half a year intervention of exercise, Mediterranean type diet, and mindfulness, stress reduction. And with all of that, I think that the future is pretty bright, and the exciting thing is what we're doing here very much connects to many other researchers here at the center, because it's so much data coming in from so many diverse different sources that the only way to truly understand this very complex system in which the brain exists, and the brain itself, of course, is to do this together. And we all need all of you to do this as well. Without you participating in research, we couldn't do what we're doing. So thank you very much.
So Silke would be happy to answer questions in the open discussion period, and I'm sure you'll have many. It's really, this type of research resonates with many of you, I'm sure. I just wanted to end the formal part of the presentation now with with letting you know that all of this research depends on funding to support the staff required, everything else that we do. And a lot of that is from grants, federal grants, provincial grants, but it can be difficult, particularly to support this interdisciplinary and sort of platform-based research, some of which you've heard about today. So if anyone is interested in supporting that, we hope you'll talk to us individually or go on our website, and there is a place where you can donate. And I'm just going to leave that there and open this up to any questions you may have about any of the presentations or anything about brain health.
There we go. Okay. I'm technologically challenged all the time. In terms of diet, I probably wasn't maybe not able to do the research based on organic versus non-organic. And like, I consider pesticides a huge problem in our environment. And if there's anything going on in that area.
Yeah, so a very good question. Is it worth paying more money for organic food or not? And I think there's certainly much more work to be done. There are some studies showing that there are higher levels of pesticides in people or lower levels of pesticides in people who eat mostly organic food. How that directly connects to things like, for example, Parkinson's, we don't necessarily know yet. But what is very interesting is that, of course, whatever comes in from the environment, and that would be any pesticides as well, and pesticides themselves, if you use high enough dosages, it can, of course, cause things like Parkinson's disease. So somebody, is farming, is using a lot of pesticides, that would be certainly a risk factor. But they need to be processed, by the microbiome when they come in. And then there are studies to show that it also depends on the host genetics, as we just heard so eloquently earlier today, our individual genetic makeup, whether we can or can't process certain things that are coming in, how harmful they are. So we need to put this all together. We need to put the input and then the different kind of processing steps in our own makeup, and I throw the microbiome in there to do this research. And I'm certainly very interested in that, but I can't say that we have a lot of results in this area yet.
I wonder how do you get their attention? Wave flag.
Hi, my name is Sarah Tambasso. Thank you so much for putting this event on. I just wanted to say that because it's incredible. I'm so happy to be here. I am a health coach. I'm called Canada's Holistic Health Coach, and I attend the Institute for Integrative Nutrition. In my studies, I've learned that they say that there's a second brain, and it's in our belly, and it's actually more powerful than the one in our head. And so personally, I was diagnosed with a traumatic brain injury, and I've applied nutrition, probiotics, and some things to completely overhaul my brain usage. My question today is, you're mentioning this gut microbiome, intestinal flora at the end of your presentation, but specifically only talking about it as it relates to Parkinson's disease. Will this studies that you are doing just as it relates to Parkinson's, will this apply to all of the other brain issues that you're dealing with, or just as it relates to Parkinson's?
Great question. Yeah, so the reason why we just picked one project is that, it's limited time. Having said that, we've done a similar project in Alzheimer's disease as well. There are colleagues, Dr. Helen Tremlett, I don't know whether she's here today, does this work in multiple sclerosis. And so there's, of course, work in autism going on. Generally, I don't do this, but the gut microbiome is of great interest there. What is interesting is that I think there are themes evolving across the board. And that certainly is that diet, nutrition plays a big role. Our gut microbiome is in all likelihood playing a role as well. We need to move from correlational studies to interventional studies to look at mechanisms. This is obviously all rather complicated. We need to look at the steps that make the communication between the gut and the brain. You know, there are many avenues for that. So I think when we do the research in Parkinson's, we can also learn certain things that would then applicable to other conditions. And so one of the research areas, I mentioned the Impact 360, where we have this combined exercise, nutrition, and mindfulness intervention. We do this for healthy ages, but we're very hopeful that we can run this for people with Parkinson's soon. This can be repeated in other groups as well. So people who've got a very early form of Parkinson's, people who live with depression, etc. And then we can compare those different groups with a very comprehensive analysis. And we also have, I'll keep that short, but we also have the BC Brain Wellness Program, which is in a way looking at all the conditions we treat at the center here and offering immediate interventions and exercise classes, creative classes, how to cope with brain disorders, very soon also cooking and nutrition classes, and related research across the board of the conditions we treat here. It's a little bit our, outreach arm. It's free. You can sign up online. There's some wait lists because it's very popular, but that speaks to our efforts very much at the center to not just look with, in a silo in one condition, but make this applicable across the board.
Hello. I'm in the top row here. Looking at the moderator lady. I don't want to cut somebody off.
It's not working.
Oh,
No,
there
it's working. Here we go.
Oh my God.
I'm not sure who this would be best addressed to, but just generally, you're talking about the importance of sleep for brain health. How would you balance with someone particularly who's perhaps had trouble sleeping their whole life, not just because of their being all elderly? What is the balance between taking sleep medications, especially if you find that they perhaps make your memory get worse or they make you feel dull or whatever? Which is better, to stay awake or to take the medication and go to sleep?
I think with sleep, you've mentioned the third kind of pillar of health. We've talked about exercise, physical activity. We've talked about diet. Sleep is that other third part. And we can approach this question from two perspectives. One is, all right, what we should all be doing and try to work towards. And if we think of health as a system, not just one thing leading to another, but rather if we increase a little bit our physical activity, if we start eating a bit better, we sleep a bit better. If we sleep a bit better, we have more energy in the next day. We can do a bit more physical activity. Maybe we're in a better mood, more energy, eat healthier. So that's the well-being approach. That applies to all of us, right? The more we can do, the better. Now, it sounds like you're posing now more of a clinical kind of question. Well, I'm doing all these things, but yet I'm not sleeping well. Sleeping well is critical. And if someone is not sleeping, it's way better to take something to sleep if it's a clinical condition that is leading to that than not sleeping at all. Less hours of sleep, strong correlation with lots of issues, from mortality to dementia, you name it. So I hope that balances the answer in a general sense. The more that we all can do on those three aspects, the better off that we are all going to be.
if there's a clinical condition that leads to difficulties with sleep, better to treat it than not treat it.
Thank you for your time. I appreciate everything I've learned here today. It's very informative. I'm inquiring about 21 years ago, my daughter had a traumatic brain injury with partial left frontal lobe removal. So years ago, I had heard the term personalized medicine. Also here, I heard it today. She struggles with a lot of her executive function, although she graduated from school, but in life now, she's suffered in so many ways. I mean, poor regulation, addictions, suicidal, like it's just all going on at the same time. It seems like right now we're just sparking in all avenues. And I'm just wondering if there's anything, I know a lot of what is here today is related to the brain research, but is there anything that's come along in the last 21 years that perhaps I don't know about as a parent of someone with a brain injury that could be recommended for her?
I will try to tackle this. So just in full disclosure, I'm not a clinician, so I don't see patients with traumatic brain injury. But what I do know that there are several major global studies that are working on the questions of how do we create the individualized programs for people recovering from a traumatic brain injury to optimize their quality of life. We're still, I would say, at some of the beginnings of this. And as somebody who started to work on neurotrauma after, and I still work on Alzheimer's disease and other conditions, I'm humbled by how challenging traumatic brain injury is because it's different for every single person. So that means understanding what works and what doesn't is very, very challenging. And this is why there are major studies in Canada, in the United States, in Australia, in Europe, that together we have enough people participating in those studies that we can hope to get some answers soon. So that's what I can share.
No, I think the person I want to address is Dr. Teresa, but she's not here. I hope the experts can help me. Now, I have been meditating, yoga. I've been doing yoga for about 65 years and meditation for the last 43 years, morning and evening. I continue with the hope at the end of my life, I'll be having a better life and everything will be normal. I'll tell you a story. A friend used to come to me and sit with me and talk to me. You want to talk to him? I am lying down. Tell me, I am talking, you're not replying. What happened? You're sleeping. He asked questions, but I am sleeping. So those are the conditions I had at some point of time. Now, for the last, I will say about 3-4 months, I am getting a feeling of, I diagnosed it as depression. Now suddenly I feel, no, I can't do this, I can't do that, I can't do that. Only things, all this helps me immediately when I feel it. I'm at work, I'm still working, I'm going for work now. And I ask for about 10 minutes, I want to go out. So they give me. I go there and do my normal exercises or yoga. And I get back to normal. 7 minutes, 8 minutes only. No cost, nothing. Only thing is 7 minutes, I am getting back. But this continues. So I consulted my physician and I was telling about this center and all that. I have read this, why don't you for me and all that. He said, no, you're expert, you're good, you're doing all that. I will take you to more than 90, 100. Don't worry, you're excellent. Be positive in your thinking, you'll be all right. Then I got this and I got this. I thought, okay, what the experts are here, I'll go and put it here and see why this is happening. Even now, every day, every other day, I go for tennis, I go for gym, I swim, and all that are happening, accordingly. But it's all coming back. Suddenly I feel it. Oh, I can't do this.
So I'm going to say, I think I got the gist of what you said. You're doing all the things that we said, or lifestyle, things that you can control. What you can't control is the genetics that you're born with, the genes that you're born with. And so all of us have different variations on that, and we're more susceptible to one thing or another. But I can probably guarantee that you have optimized everything you can optimize, and more power to you, and keep going. And also, I think this sounds a little, cheesy, I suppose, but positive attitude makes a big difference too. And just, just making the best of what you can do and not dwelling on the limitations, because as we age, we will all have some. So thank you for sharing that. We have a lot of questions here. We'll go with you.
Thank you all. This has been super interesting, but this is a question about Alzheimer's. Just wondering, we hear about, amyloid proteins and tau proteins, and I'm just wondering if are those still of interest within study, or are there any other new proteins or molecules that have come up that are interesting for research? Thanks.
I will take this question. So thank you so much. There is such exciting and hopeful interest in Alzheimer's disease right now. Last year, there was a new drug that was approved in the United States that is an antibody that can pull amyloid out of the brain. And that works quite well to get rid of the amyloid. The patients who were on that drug had about a 25% slower rate of cognitive decline. So the anti-amyloid therapies, there are now 2 that are approved, but neither are yet available in Canada. And there's still a lot of research that needs to be done on, can we get better than 25%? Maybe if we started a little bit earlier, for example. There are certainly really amazing interest in early detection of Alzheimer's disease. So by the time people develop the memory problems, we know that they've had brain changes maybe for 15, 20 years already. And what we're finding is that there are certain kinds of blood tests, again, back to that can measure, that can detect these changes that are happening maybe 20 years earlier. So in a person's 50s, for example. So the world is now shifting into trying to look at potential therapeutic or interventional approaches before a person actually develops the memory problems. So absolutely, tau is one of the major markers that appears to be correlated with early detection, and anti-tau therapies are certainly being developed as well and tested. But it's the anti-amyloid ones that are now clinically approved, and we just need to do some additional research to have those drugs available in Canada, or identify less costly ways to achieve the same benefit. But it's a watershed moment for Alzheimer's disease and dementia, because we are on the cusp of, number one, having effective disease modifying therapies, and number two, being able to maybe detect the very beginning parts of the disease in time to prevent the cognitive decline. So that's so exciting.
Yeah, I completely agree. And I think one of the areas with which there's a lot of hope in the future is through these new technologies that are being developed right now. So we've been talking a lot about sequencing primarily from genomics, but now we're beginning to grasp, what are the first cells that show manifestations of various types of Alzheimer's pathologies, and understanding how those cells are changing and what we can target within those cells. So this is really moving us as a community towards prevention. If we can understand what's initially affected, we can target that specifically. And also, in addition to using or pursuing these known candidates, we can hopefully discover new drug targets, and through that develop new therapies that are, would have been previously unforeseen.
I think there was one right here. Yeah.
Hi, good morning. My name's Elizabeth Bonner. I'm with Afterstroke BC, and I just want to say thank you to all of you. First of all, certainly all of our clients, or the vast majority of them, have benefited from BC Brain Wellness. But I'm going to put on a slightly different hat, more at a personal level. And I guess my question is for Dr. Fidel, Vila-Rodriguez. In the mid-70s, my mother was dealing with what in all likelihood now would have been, or what we'd refer to as postpartum depression, and received electroshock therapy and rather draconian process or procedure. And I'm just curious, with the TMS, is there brain or cell death, or what is happening in that procedure?
Absolutely. Again, we have the analogy, right, of electricity, and we kind of thoughts about ECT versus TMS come up all the time. The amount of energy and the current that is induced in the brain with TMS is very focal, very small, and it does not get anywhere close to, and we don't want to trigger a seizure. And the mechanism and the way the TMS works is stimulates a small area, and that becomes the entry point, meaning we are understanding now the brain as a very complex network where things are very connected to each other. And when we stimulate a small area in the cortex, in the outer part of the brain, that through connectivity can also modulate brain activity in other parts of the brain that might regulate level of energy, decrease suicidal ideation, increase appetite. So that's the way that TMS works. It's like sometimes I use the analogy of an external pacemaker. We are giving a rhythm to the brain, and we are tapping on brain circuits. And so that's more like how TMS works.
Thank you.
And it's been used also in the context of reproductive psychiatry. So there's a few studies in people pregnant, and so as opposed to medications.
So some of us have seen the movie Concussion, and I'm interested in CTE, and if UBC or your institution is looking at CTE study in the future. As we know, CTE now from a study perspective is postmortem, and I'm really interested to know if there's going to be perhaps some developments in premortem analysis. People in my vintage who played football and rugby as a child in the 60s and then played at a higher level in the 70s. Some of us are, and many of my teammates and colleagues over the years, we suffered, some of us 10 or more substantial concussive events. And I'm okay, I'm here, but the questions that my family would have is, has there been some degradation in my situation over time? I don't feel I'm in any trouble right now, but there's a lot of players, athletes around the world who are being concussed in all kinds of sports that didn't used to have them, that now it's happening. So that's my question is there going to be some activity in CTE at UBC? My brain is going postmortem to University of Toronto right now, but if there was a program here, I would shift it.
Thank you so much for that question. Certainly, UBC is involved in many studies of concussion, CTE, and outcomes of traumatic brain injury. This is largely being coordinated at the national level. So UBC is one of the major sites that have enrolled for our national study called CAN-TBI. So that takes individuals with traumatic brain injury from the acute stage and looks further. You're correct. I think that the best and most experienced neuropathologist looking at CTE is Liu-Ambrose, who is at the Hospital for Sick Children in Toronto. And so I think your brain is going to a great place. We have awesome neuropathologists here, but I think Liu-Ambrose is probably the most experienced at looking at CTE. And what's really interesting about her findings is that she has, I think, the largest collection of repetitive concussed brains in Canada. And she's finding slightly different things than what have been reported from Anne McKee and her colleagues about what CTE might be looking at. It's a fantastic program and opportunity. Now, where the gaps still lie are individuals exactly like you have had multiple concussions in the past, are curious about how you're doing. It is actually really hard to design and get funded studies that are not starting right from the moment of injury and coming up. So this is where it would be fantastic to have more resources available so that we can get more studies and enroll more participants who have a history of concussion to see what is really happening.
Thank you.
You have time for one more question. That would be you, and then we're going to wrap it up.
There. Now it's on. Alzheimer's is probably one of the toughest things to crack. And I unfortunately have that in the early side. Now, there's a lot of research going on around the world and here, which is wonderful. But to crack that problem. But in the meantime, there has been some considerable research in North America and elsewhere about photobiomodulation, which has shown us the ability to slow down and perhaps even in the future stop it in its steps, so that people can live a proper life before it gets worse. And I'm wondering if any research of that sort is being done here.
It's a great question. To clarify, the photobiomodulation is using things like light stimulus to drive and entrain brain activity and then clear things like plaques. You're right. So that research, it was it looked completely transformative when it was initially released. There was a lot of hope that was driven by that study. The general idea behind this is if one drives the brain with things like light at a particular frequency, you can begin to clear out signatures of the brain associated with Alzheimer's. That work has been, that particular experimental paradigm has been looked at by other research groups, and the effects have been mixed at this point. And as a result, the clinical relevance and efficacy is still to be determined. It would be absolutely wonderful if this were an alternative strategy, and if it were to be, it would be outstanding and could be coupled with pharmacologic interventions and other types of interventions as well. But the ultimate, the utility of that assay in particular still needs to be rigorously studied, unfortunately. It looked hopeful at first, but now we don't have the same signatures as in the original study.
Well, thank you so much for all your great questions, for your attention and patience. It's kind of a long program. There's some people who might need a little bit more time to get out to get their food. So if you aren't one of those people, maybe let them go ahead, and we have a movie or a video we can show you about the DMCBH. It only takes like 3 minutes. So we'll do that now. But if you're in a rush, go ahead out now.
1 in 3 Canadians will have a neurological or mental health condition in their lifetime, making brain disorders a leading cause of disability in Canada. Understanding how the brain works is one of the biggest undertakings of our time. The Djavad Mowafaghian Centre for Brain Health at the University of British Columbia is at the forefront of brain research and patient care in Canada. World-renowned medical care is provided at 5 specialized clinics and through clinical trials that examine how effective and safe new therapies and medications are. Medical breakthroughs are possible because of research happening at our center. the first step in developing treatments is understanding how the brain works at the genetic, molecular, and circuit level. Only by understanding how the brain develops, ages, and functions can we diagnose, prevent, and treat brain disorders and injuries. There are over 100 clinical and foundational researchers at the center, working together to bridge research and healthcare. The sensory motor team is focused on understanding how the brain controls movement and processes sensory information to develop better treatments for disorders like multiple sclerosis, Huntington, and Parkinson disease. Other scientists study injuries that happen to the brain, like a stroke or traumatic brain injury, and look at how the brain can be repaired. Our mental health and addictions group is paving the way towards developing effective new treatments for disorders like depression and anxiety. Researchers focused on learning, memory, and dementias aim to discover how the brain stores information and how this is affected when a person develops disorders such as Alzheimer's disease. Another group of researchers aims to determine how the brain develops from infancy to adulthood and how disruptions in brain development can lead to autism, epilepsy, and intellectual disabilities. All of this research is supported by our many state-of-the-art facilities, like a cryo-electron microscopy facility, which uses microscopes up to 13 feet tall to take pictures at near atomic resolution. A biobank, where human tissues and samples are stored. A neurocomputation and preclinical imaging center, which contains high-performance microscopy and computing equipment. And a neuroimaging suite, which houses advanced PET and MRI scanners used to study the human brain. At the heart of what we do at the center are our trainees, who are being mentored as the next generation of scientists. By uniting research, education, and clinical care, the center is a hub for brain health and well-being. As we unlock the mysteries of the brain, we will be able to develop more effective therapies against disease and improve the health of all Canadians. By working together, we can continue to advance brain health across the lifespan.
Okay, well, feel free to stay and get something to eat outside in the atrium, and chat with us if you have more questions. For those of you on a tour, we're probably going to start at about 12:30. So you'll have signage to match up with your tour guide. Thank you so much for coming.